10.2 Mitosis. Challenge. Using the diagram of a football field, write a short paragraph describing how the players, the midfield line, and the goalposts compare to the structures of a cell involved in mitosis and cell division. The players represent the chromosomes, the midfield line represents the equator of the cell, and the goalposts represent the centrioles at the poles of the cell. Objectives: what are the phases of the eukaryotic cell cycle? What are the 4 stages of mitosis? How does cytokinesis occur? Eukaryotic cell cycle. The cell cycle is a repeating sequence of cellular growth and division during the life of a cell. The life of a eukaryotic cell cycles through phases of growth, DNA replication, preparation for cell division, and division of the nucleus and cytoplasm. The cell cycle is made up of 5 phases. The first 3 phases together are known as interphase. The remaining 2 phases make up cell division. Cell cycle. Eukaryotic cell cycle interphase. During interphase, the cell is not dividing. It is growing and preparing to divide. Different types of cells spend different amounts of time in interphase. Cells that divide often, such as skin cells, spend less time in interphase. Cells that divide seldom, such as nerve cells, spend most of their time in interphase. Eukaryotic cell cycle interphase. During the first gap phase, G1, a cell grows rapidly as the cell builds more organelles. For most organisms, this phase occupies the major portion of the cell's life. During the synthesis phase, S, a cell's DNA is copied. At the end of the S phase, the cell's nucleus has twice as much DNA as it did in the G1 phase. During the second gap phase, G2, the cell continues to grow and prepares to divide. Hollow protein fibers called microtubules are organized in the cytoplasm during G2. Checkpoint. What is the cell cycle? What takes place during the cycle in eukaryotes? How many phases make up the cell cycle? What are their names? What are the 3 phases that make up interphase? What happens in each phase? Eukaryotic cell cycle cell division. Each new cell requires a complete set of organelles, including a nucleus. The process of dividing the nucleus into 2 daughter nuclei is called mitosis. The process of separating the organelles and the cytoplasm is called cytokinesis. Eukaryotic cell cycle cell division. During mitosis, the nucleus divides to form 2 nuclei. Each nucleus contains a complete set of the cell's chromosomes. The nuclear membrane breaks down briefly. The 2 sister chromatids of each chromosome are pulled to the opposite sides of the dividing cell. Eukaryotic cell cycle cell division. As the nucleus divides, the cytoplasm also begins to divide. Each daughter cell receives about half of the original cell's organelles. During cytokinesis, the 2 daughter cells are physically separated. Checkpoint. What is the difference between mitosis and cytokinesis? Summarize the events of cell division. What is the end product? Stages of mitosis. Stage 1 prophase. Within the nucleus, chromosomes begin to condense and become visible under a light microscope. The nuclear membrane breaks down. Outside the nucleus, a special structure called the spindle forms. The spindle is made up of several spindle fibers. Stages of mitosis. Stage 1 prophase. Cells have an organelle called the centrosome, which helps assemble the spindle. In animal cells, the centrosome includes a pair of centrioles. Before mitosis, the cell's centrosome is duplicated. During prophase, the centrosomes move to opposite poles of the cell. Prophase. C. B. F. E. A. D. Stages of mitosis. Stage 2 metaphase. During metaphase, the chromosomes are packaged into their most condensed form. The nuclear membrane is fully dissolved, and the condensed chromosomes move to the center of the cell and line up along the cell's equator. Spindle fibers form a link between the poles and the centromere of each chromosome. Checkpoint. What is the first phase of mitosis? What happens during this phase? What is the second phase of mitosis? Stages of mitosis. Stage 3. Anaphase. Once all of the chromosomes are lined up, the spindle fibers shorten. Sister chromatids move toward opposite poles as the spindle fibers that are attached continue to shorten. Each pole now has a full set of chromosomes. Stages of mitosis. Stage 4. Telophase. A nuclear envelope forms around the chromosomes at each pole of the cell. Chromosomes, now at opposite poles, uncoil and change back to their original chromatin form. The spindle dissolves and the spindle fibers break down and disappear. Mitosis is complete. Checkpoint. What is the third phase of mitosis? What happens during this phase? What is the last phase of mitosis? Cytokinesis. As mitosis ends, cytokinesis begins. The cytoplasm is separated, and two cells are formed. During cytokinesis, the cell membrane grows into the center of the cell and divides it into two daughter cells of equal size. Each daughter cell has about half of the parent's cytoplasm and organelles. The end result of mitosis and cytokinesis is two genetically identical cells in place of the original cell. Cytokinesis separating the cytoplasm. Vesicles holding cell wall material line up across the middle of the cell. These vesicles fuse to form a large, membrane-bound cell wall called the cell plate. When it is completely formed, the cell plate separates the plant cell into two new plant cells. Cytokinesis continuing the cell cycle. After cytokinesis is complete, each cell enters the G1 stage of interphase. The daughter cells are about equal in size, about half the size of the original cell. The activity of each cell continues because each has its own DNA and organelles. The cell cycle continues for each new cell. Checkpoint. When does cytokinesis begin? Cytokinesis divides the cell contents in underscore. What is the result of this process? How does cytokinesis in plants differ from animals? After cytokinesis, what phase does the new cells enter?